Good evening and a virtual hello to everyone. I'm Rob Johnson, former board member of Brain Research Foundation and eternal BRF supporter, and I am delighted to be here on the stage with my good friend, the Executive Director and CEO of Brain Research Foundation, Dr. Terry Constantine. It's great to be back as the MC of BRF's annual event. I've had this role for many years and have always enjoyed it for a couple of reasons. First, BRF is a remarkable organization making an important difference in the lives of many. We'll hear more about that shortly. And second, besides being a great cause, BRF is also made up of great people, talented and skillful people who are deeply committed to do what they can to help others. While this year's event is obviously different and we can't be together in person, I'm still happy to join you virtually in your homes. So sit back, relax, maybe pour yourself a beverage, get into comfy clothes if you haven't already, and remember, we can't see you, and get ready to enjoy a very interesting program. Now, before we move on, I want to mention our co-chairs of the evening, Scott and Mary Sirota. Facing the unique challenges that our new world has presented, Scott and Mary have done a wonderful job of navigating us to where we are tonight. I would also be remiss if I didn't mention the Foundation's outstanding board chair, Peter Pond, for all of his efforts to make this event a success. And my friend, Norm Bobbins, who every year seems to step up to the plate and do everything he can to help BRF. And to all the wonderful supporters of BRF, thank you for your commitment to BRF and its work to fund much needed research that gives us a better understanding of brain function, disorders, and injuries. With everything going on around us, the stakes are higher than they have ever been before. Innovative brain research is essential, and BRF simply cannot do what they're doing to advance this vital research without your support. BRF needs you on this team. You will see on your screen a way to help, the brf.org/to donate. We understand that many of you have already given, and please know it is appreciated. But if you can find it in your hearts to help again, it would mean so much. And if you have yet to give, we know that these decisions are difficult, especially during these times. But if you're able, we would love to have you with us. No donation is too big or too small. Every dollar counts. Now, as we continue with the program, I realized that earlier I failed to mention another thing that I love about this event. This has always been an evening that I left feeling a bit smarter and a bit more enlightened, an evening where I actually have learned something. And I assure you that tradition will continue to live and thrive with our panel discussion later in the program. This year, we welcome 3 distinguished experts on autism. And facilitating the conversation will be Dr. Terry Constantine. Many of you are familiar with Terry and the extraordinary job she has done at the helm of BRF. Terry's dedication to BRF stems from the fact that she is a scientist herself and is acutely aware of the importance of research, as well as the funding obstacles that scientists continuously seem to face. So let's hear from her right now. Ladies and gentlemen, Terry Constantine. Terry.
Thank you, Rob. And thank you for being such a great friend to Brain Research Foundation. Since 1953, BRF has supported cutting-edge neuroscience research. And in the 60s, our slogan was, "No family is left untouched." This is still true today, 68 years later. Unfortunately, everyone knows or has known someone struggling with a brain-related issue like Alzheimer's, addiction, and depression. And even COVID-19 has neurological symptoms tied to it, like loss of smell and taste, and even an increase in strokes. Brain problems have confronted us or will confront all of us at some point. Brain cancer, more specifically an aggressive glioblastoma, took my mom at the age of 70. It is something that I wish no one ever has to witness themselves. But that experience made me even more passionate about this foundation and our mission. We need discoveries, we need new treatments, and we need cures. How do we do that? Simply put, we find the most innovative neuroscience research projects in the nation, and we fund them with seed money. It's similar to being venture capitalists. We invest small amounts of startup money into new and brilliant ideas that scientists propose so they can get the initial data and results they need to catapult their research into larger projects funded by NIH. And how do we know that we are successful? We know this by carefully measuring our return on investment, by assessing how much money is raised after our initial seed investment. We can say that every dollar BRF awards, an average of an additional $25 is awarded to the scientist to continue their research. By any standards, this is an extraordinary return on investment. None of this funding is possible without generous contributions of our donors. And I want to thank all of you watching who supported our first virtual discovery event. Without you, we would not be able to continue our mission. Thank you to my fellow Board of Trustees and our young leadership board members. I could not ask for a better group of ambassadors for BRF. Thank you all. Finally, let me tell you who is responsible for this wonderful event. Tonight's co-chairs, our Board of Trustee member Scott Sirota, along with his wife Mary. Thank you for taking on this very important job and creating such a successful event in such an unusual year. I'm very pleased to turn the program over to Scott, who will be telling you a little bit more about why it is so important to them to support BRF. Scott?
Thank you for that kind introduction, Terry. And thank you for the terrific work you do in leading the BRF's important work. As Terry said, I am Scott Sirota. I'm a member of the BRF Board of Trustees and the president and CEO of the Blue Cross Blue Shield Association, at least for the next 2 months before I retire. My wife, Mary, and I are honored to be co-chairs of tonight's benefit. Thank you all for joining us in these truly extraordinary circumstances. I believe it's safe to say that everyone tuning in tonight has at some point known someone struggling with a brain-related issue. Whether it's Alzheimer's, mental illness, autism, or injury, unfortunately, brain issues are or will be a reality for most of us, either for ourselves or for someone that we love. Someone that I love very much, my grandson Ethan, is thriving and growing up with autism. Ethan is an amazing person, and just like I do for all my grandkids, I wish and pray every day that he will lead a happy, healthy, full life, his best life. Frankly, he's one important reason why I decided to be part of this organization. Another reason is its focus on finding and nurturing innovative solutions. My long career has consistently reinforced for me the importance of innovation. Breakthroughs in healthcare, like in most other arenas, start with innovative thinking, seeing and attacking problems differently, experimenting to see what works. The BRF truly values innovation when deciding which research projects to fund. More traditional funders view many projects as too speculative. But BRF believes at its core, there is high potential to yield important insights by investing in such promising science. That's why in 2017, the Blue Cross Blue Shield Association partnered with the foundation to help tackle the country's opioid crisis. BRF formed a committee that pursued an innovative approach. They would focus on research aimed at reducing relapse. Nearly 90% of those who seek treatment for opioid use disorder end up relapsing. So there is an urgent need to expand relapse prevention therapeutics. One funded scientist looked at repurposing a drug commonly used to treat depression. Another pursued research in the nasal delivery of a drug that increases the survival of cells threatened by addiction. These innovative projects are still underway and hopefully one day will provide a treatment for those who struggle and will provide relief to the family members who are desperate to help their loved ones. A third reason why I joined this board is because of what the foundation offers to researchers just starting out in their careers. I'm very passionate about mentoring and helping develop the next generation of leaders. The Brain Research Foundation does the same thing with their young investigators by funding new labs and helping talented researchers explore their fresh ideas that can lead to discoveries and the starting point to new treatments and cures. The funding provided because of your support enables today's breakthroughs while simultaneously laying the foundation for the discoveries of the future. In closing, I want to thank my fellow Board of Trustees, our dedicated supporters, and new friends joining us tonight. If you are new to us, I encourage you to learn more by visiting our website. And please consider making a donation that will enable this terrific organization to continue its very important work. Thank you, and enjoy the rest of the program.
Thank you, Scott. And thanks also for your important partnership with BRF. As Scott mentioned, BRF is at the forefront of innovation with a commitment to explore novel ideas that lead to discoveries that will ultimately help us all. These discoveries don't happen by accident. Hard work, brilliant minds, and generous donors are the key. BRF and the innovative research they support needs your support as well. We need you to be part of the critical foundation that builds ideas and discoveries which make a positive difference in the lives of people challenged by neurological injuries and disease. We hope you will join us in this vital cause by going to thebrf.org/donate. It will mean a lot to have you by our side. Now, another thing Scott mentioned was the importance of mentoring the next generation of leaders, and we are so fortunate to hear from one tonight. Ladies and gentlemen, Lauren Shapiro-Mandel, co-chair of Brain Research Foundation's Young Leadership Board. Lauren.
Thank you, Rob. Hello, everyone. As Rob stated, my name is Lauren Mandel, co-chair of Brain Research Foundation's Young Leadership Board. Founded in 2017, our dynamic group of young professionals focuses on our mission to promote, raise awareness, and raise funds for Brain Research Foundation. My personal story is what initially brought me to BRF. My two co-chairs, Joe and John, also happen to be my brothers. Our mother, Rhonda, suffered a cavernous angioma, a type of brain bleed, when I was only in middle school. And this past June, she passed away after fighting the effects of her disease for more than 20 years. Of course, her life was by far the most changed as a result of her illness. But like so many other families, our mom's illness also changed and shaped our lives in many profound ways. While it may seem like brain-related disorders are far from our young minds, this could not be further from the truth. Every single person on the Young Leadership Board has already been touched by brain disorders. Some face their own struggles with ADHD, OCD, or anxiety, while others support family members fighting Parkinson's or brain tumors. Many of us are starting families of our own, and we worry about diseases like autism, while also watching our parents' generation start to feel the cognitive effects of aging. There is no escaping the many, many ways brain disorders affect us all, even at a young age. I know firsthand why it's important to act now, rather than waiting for a family member or friend to reach a state of devastation. Now is the time for us to continue to build a passionate board of individuals who are dedicated to funding research for all brain disorders. Since 2017, our group has raised over $200,000, which has been used to help fund BRF's seed grant program. In fact, just last year, we funded 1 year of a seed grant for Dr. Megan Williams at the University of Utah, which we hope will help answer questions related to epilepsy, mental illness, autism, and intellectual disability. I would like to thank my fellow board members for their passion and dedication. And if anyone is interested in joining the Young Leadership Board, please visit BRF's website.
Thank you so much, Lauren. You and the Young Leadership Board are the key to moving BRF forward. Now let's take a quick look at this video on how BRF is moving research forward.
For more than 60 years, Brain Research Foundation has shaped the future of neuroscience by saying yes to innovation, by surveying the scientific landscape to identify novel ideas that offer limitless potential to transform our knowledge of the brain. We work tirelessly to find ideas that will move science forward, brilliant ideas that demand exploration, analysis, and investment.
The Brain Research Foundation is what you would perhaps call a small organization, but with large reach. And we need to keep growing that reach. We fund scientists across the nation. We'd love to fund more. We're very nimble, and we can react to what we think is the best innovative research.
Typically, these young scientists have very bright, state-of-the-art ideas, but no money. We give them the starting capital to fulfill their research dreams and to get the NIH monies and to get them on their way.
We say yes to investigation. Researchers working throughout the country submit proposals for cutting-edge ideas that will set us on a path to finding answers to some of science's most perplexing problems.
We understand the need for this brain research, and we've expanded to universities throughout America in order to get the best and brightest.
We say yes to ideas that generate results, results that demand even more work and more investment. work that draws the attention of a community of scientists focused on ways to improve brain health.
I think that the BRF is critical for neuroscience research. It lets us do those initial, get that initial preliminary data that allows us then to get grants. So it's a really critical organization.
Our research extends to every neuroscientist conducting novel research in the U.S., to every laboratory and every research institution in the country. Dr. Chetkovich leveraged a BRF grant into a multi-million-dollar investigation into an epilepsy gene therapy treatment that may be as few as 5 years from human trials.
Translating the studies that we've done with the BRF money into clinical trial is our goal.
For more than 60 years, the Brain Research Foundation has accelerated the future of neuroscience research with a simple yes. I want to thank you all for being part of that journey, and I hope you continue along with me as we move forward.
Say yes. It is a simple but meaningful message. And now, as promised, one of the highlights every year, our panel discussion, Autism Research, Genes to Behavior. And to moderate the discussion, once again, Dr. Terry Constantine. Terry.
Thank you again, Rob. I would like to introduce our three panelists, all of whom are well-known researchers in the field of autism, and all of them have personal connections to the research they are so passionate about. Dr. Ted Abel from the University of Iowa. Dr. Abel was a longtime member of the BRF Scientific Review Committee, which reviews and selects the grants BRF funds. Dr. Ed Cook from the University of Illinois at Chicago. Dr. Cook is one of BRF's greatest success stories. In the 1990s, Ed received a few small seed grants totaling less than $50,000. With the data from these small grants, he was able to obtain almost $15 million to continue his research. And Dr. Suma Jacob from the University of Minnesota. Dr. Jacob was mentioned in a past BRF annual report by Dr. Cook. He trained her at the University of Chicago, and then her career took off. Thank you all for being part of this discussion. First, let's define what we're talking about this evening. Ed, what is autism spectrum disorder?
Well, first of all, let's talk about what a spectrum disorder is. It means that there's no clear cutoff of where autism begins and ends. And so I'm going to refer to autism just to make it easier. So autism is a combination of two major problems. One is difficulty in social communication, and the other is restricted and repetitive behaviors. Social and communication difficulties. You could think of an early one in which obviously babies have eye contact with their caregivers. That would be an example of one that would be present early in development. And then you can also think of someone who's an adult with otherwise normal cognitive function who has difficulty, let's say, with subtleties of connecting language with nonverbal communication and conversation. And for restricted and repetitive behaviors, one example that we used to talk about a lot that you might see with younger children would be, instead of playing with a toy truck in the usual way, being focused on watching the spinning wheels would be an example. And we've also been interested in other examples where routines have to be followed, or someone might get very upset.
And so, Suma, let me throw it over to you about diagnosis. It seems like Ed touched upon some things that you're looking for, but obviously you can't see all of those things in a doctor's office. What are you specifically looking for to make a diagnosis?
So clinicians look at the child's developmental history and also look at behavior. And we have some standardized behavioral tests as well as interviews where we can get those detailed information about the different things like social communication and rigid or flexible or repetitive behaviors that Ed mentioned. And we have been getting better and better at doing this earlier and earlier in childhood.
And Suma, what is the prevalence of autism?
So the current prevalence is 1 in 54, and the CDC has been updating this every handful of years, and this is the newest number that we are going by.
And Ted, let me ask you, are there treatments for autism? And if so, what are they?
One of the most important things about autism is early identification, identifying individuals as early infants, as early as we can, and there's been much progress on that. There's no sort of silver, no bullet here that can fix autism, and in many ways, there's aspects of autism that in some individuals can make them do certain jobs, for example, better than typically developing individuals. But there are treatments like cognitive behavioral therapy and more behavioral management therapy, and those involve really time-intensive work, applied behavioral analysis to understand why individuals or infants are doing what they're doing in response to specific stimuli. And those treatments are often in the context of school. They often begin in kindergarten and pre-kindergarten, and they continue in a school setting. It's also worth noting that individuals with autism often have other medical challenges and issues. And so a broad medical management, managing other issues like issues with their GI tract or asthma or allergies, other issues that can affect their performance. But ongoing therapy, even into adulthood, of speech and language and occupational therapy continues to be important. Our son Seamus is on the autism spectrum, and he's now 20 years old. He's literally had therapy since he was 4 days old. He was in NICU as a 4-day-old, and he had a physical therapy then, which was largely massage at that point. And now he has an occupational therapist to help him navigate school. He's a very high functioning and currently a freshman at the University of Iowa, but has never had a friend. As Ed said earlier, that you can see the difficulty in social interaction. The most important thing, though, beyond these effective treatments and interventions, is really that we begin to accept individuals with autism. We've been working towards awareness, to be aware of autism spectrum disorder, but the critical difference will come when we accept them as part of our society and part of our daily lives. That will be the most effective therapy.
Well, and I think I would follow up on this. If you are not connected with someone personally that has autism, you think of autism as a childhood disease, but they grow up. And that's where this acceptance is so important as they're moving through society. I think that's what you're trying to say, Ted, and I totally understand and agree.
Right, and understanding really how these individuals can respond under some circumstances that we would find shocking, and that there's been some interactions with individuals with autism and the police, which are really terrible and terribly disheartening. There was an example of a disorder of an individual with Tourette syndrome, not autism, but that was kicked out of a university recently because of their tics that they're unable to control. And so understanding what others are going through is really the most critical thing. And that's really what I think I've learned the most from Seamus, is seeing his perspective on things and the challenges that we would otherwise think, well, this is just a normal situation, but he can't handle the background noise or other aspects of it that make it more difficult for him.
And so when Ed described what the disorder was, obviously we see it, it kind of runs the spectrum, right? And like many other disorders and diseases, ASD is not straightforward. There are many factors coming into play. Way back in the 1940s, it was the parents' problem. It was they were bad parents, cold parents. We've come a long way from then, and we're doing a lot of research, but a lot more needs to be done and more to learn. Let's talk about risk factors, because I think this gives us a nice segue into the research areas. So Suma, could you tell us what the risk factors are that we know today?
So many of us know that there are genetic risk factors. So in terms of heritability, autism is one of the highest heritable disorders. And beyond that, there are issues related to genetics. So with genetics, everything that happens in early development, as well as everything that leads up to that time, can have an influence. So there have been studies looking at parental age can increase risk of autism. Other factors, including early, preterm complications, low birth weight, complications in maternal health, all those things that happen as an infant is developing can be involved and affect risk for autism as well.
So let's start with genetics. And I'm going to start with you, Ed. You've been working in the field for 30 years. Is it true that there are over 100 genes that have been reported to be associated with ASD? And what does that actually mean, associated?
Yes, that's true. These genes are implicated by many individuals with autism having mutations in these genes not found in their parents. The number has increased greatly with the development of sequencing of the protein coding regions of genes on a large scale. That's been all these studies started in the research labs, weren't possible. They're actually an extension of the sequencing of the human genome about 20 years ago, and now are finding their way into the clinical arena.
And a lot of times when we start talking about genes and genetics, we think that it means it's inherited. Is ASD inherited?
So as Dr. Jacob mentioned earlier, about 50 to 90% of the risk for ASD is inherited. The paradox is those 100 genes I mentioned account only for about, people would say 10 to 20% of autism cases. Now, those are genetic, but not inherited because the parents didn't have them. So much of the risk that's inherited is polygenic, meaning that hundreds or thousands of subtle variations in genes or around genes contribute to both risk and protection from autism. One interesting development that we'll watch over the next several years is something called a polygenic risk score, in which one adds up all of those subtle risk factors to determine whether an individual carries more or less of this inherited risk. But one reason for so many of these de novo out of the blue mutations is that many people with ASD do not have children. So in that sort of out of the blue mutation, the individual with autism may be less likely to have children of their own. So you wouldn't see the kind of inheritance. A lot of people think about where you see full autism in the parent and the child. That's less likely in this condition.
And did you say that they're doing this test now, the scoring, or no?
The scoring is done on a research basis.
Okay.
We just collaborated with colleagues at Vanderbilt and Northwestern, for example, to correlate what's been described as a broader autism phenotype that some, but certainly not all, and probably not even half of parents have some aspects of autism, and looking at the relationship between those polygenic risk scores and the manifestations in parents. And although there are some interesting relationships, they are not strong. And so I think we're at the early stages of polygenic risk scores. Right now, they don't have a clinical utility, for example, but they're an example of some of the research that's going on. And one reason I mentioned that study is it's exciting in terms of collaboration of the leaders in looking at subtleties in clinical manifestations and geneticists working on things like polygenic risk scores.
Okay. Thank
One thing,
you.
if I could just add, Terry, that's really important in this context is having a large number of subjects in these studies, a huge number, tens of thousands. And so one of the things that each of the families with autism can contribute here is to be involved in these genetic studies, to be involved in other studies. There's a large study called SPARK, funded by the Simons Foundation Autism Research Initiative, aiming to get 50,000 individuals, families and individuals on the spectrum. I'm part of SPARK as a parent, so is Seamus. And it's really been terrific to see everything that's been done to identify these genes. And now SPARK is also setting up a program called Research Match, where individuals and families that have been studied genetically can now be part of studies like the one Ed mentioned, to look at broader autism phenotype or look at other aspects of behavior. One example is sleep, for example. You could look with a survey at sleep differences and difficulties in individuals with autism, and then see how that was connected to this polygenic risk score that Ed mentioned.
Let's talk a little bit more about research, and I'm going to bring up a risk factor. I'm not sure you mentioned, Suma, and I think I have it correct, that there's a male bias over females. I think it's 1 to 4. And Ted, you have some recent work on mouse models. Can you talk a little bit about that?
Yeah, so our work on mouse models actually was carried out by one of Suma's colleagues at the University of Minnesota, Nicola Grisham, who was a postdoc with me, and a large number of people funded by the Simons Foundation. But what our work built on was all these genes that have been identified. That was one piece of it. The other piece was a lot of the tools, some of which have been funded by the Brain Research Foundation, to image neural circuits, study molecular mechanisms, and identify behavioral changes. And so we've done those studies in several mouse models for the study of autism. You'll notice I'm being careful to not say mouse models of autism, because we can't have a mouse with autism. But we can study things like repetitive behaviors, social behaviors, other things, to see what goes awry in mice that lack genes that are linked to autism. So we did that broadly, looking at behavior. And what was striking is we found these behavioral differences that were only found in male mice that lacked these genes. And those behaviors were changes in locomotor activity, particularly hyperactivity, and changes in the ability to work for rewards. And that's an interesting concept, because it's underlied by a circuit called the corticostriatal circuit, which is a sort of big word, but that's the circuit that mediates language learning, language production, locomotive behavior, and in particular, our ability as humans to predict what will happen next, to know that if we do this, then that might happen. And conceptually, you can think about autism as a disorder of prediction. And a lot of what individuals with autism may do that show up as symptoms, like restricted interest, repetitive behaviors, difficulty with social interactions, may reflect that challenges that they have in predicting what events are going to happen next. And that may specifically affect these particular circuits in the brain that are altered in males and that develop differently in males and females. So this basic research, this fundamental research that the Brain Research Foundation has made possible, really has enabled us to get an insight from studying mice and what they do, hopefully gain insight into a conceptual idea about autism and how it may actually be underlined in neural circuits in the brain.
Thank you, Ted. Suma, let's talk about your research. Your lab examines effects of novel interventions on social functioning and repetitive disorders. I think one of the things you're looking at is intranasal oxytocin. Can you tell me a little bit more about that and why oxytocin?
So we have completed a study of intranasal oxytocin, and then there have been other studies also done. And the reason we were interested in oxytocin is it's a neurohormone that influences social behaviors in many mammals. And so one of the things we wanted to do is study something that has not been studied before. And one of the nice things about oxytocin and intranasal is it doesn't have a lot of side effects. One of the challenges of studying things like neurohormones, and I have colleagues who are studying vasopressin, which is closely related to oxytocin at Stanford, are that these are neuromodulators, meaning, and that's sort of a big word to say, that they affect how the brain processes social information. And when we look at things for medications, we often have rating scales or symptom targets. And so far, oxytocin does not seem to move those things. In a study I did with Dr. Anagnostou in Toronto, we found that oxytocin changed the quality of life, but not some of these symptom targets. And it also changed how faces are detected as similar or different. So even though intranasal oxytocin can affect these subtle behaviors, there's not evidence for it to be a treatment in the way we traditionally look at drugs in psychiatric disorders.
I mean, we looked at 3 different labs right now and what you're doing in autism. So there's a lot of work going on. I would like to ask, in closing, each one of you, where do you think the biggest breakthrough is going to come from? Is it a combination of things? Do you see one thing on the horizon sooner than later? Ed, let me ask you that question first.
Well, I'll go back and I want to acknowledge Brain Research Foundation's assistance in this and go back to almost 30 years ago when pilot awards are so important because people were going, well, why are you studying genetics and autism? You're not going to get anywhere. And now we've got over 100, and within the next few years, with the large samples that Professor Abel mentioned, we're going to have more, and that's very exciting. And we're in the midst, and this is extremely important, of figuring out what all these genes do. And that's the critical next step. And that's something that Dr. Abel's involved in and Dr. Jacob is involved in. The other side of that is once we develop new treatments out of the lab, we're going to have to test them extremely carefully, not as simple as, if we give this, does somebody immediately have better social function or are more flexible? And so we've delivered a large number of molecules that contribute to ASD, and now is very much the stage of taking that information to basic understanding of the brain and translating it into treatments.
And Ted, do you have something to add? Do you agree?
I very much agree. I think I would add 2 things. One is on the fundamental science side. I've already mentioned the importance of organizations like the Brain Research Foundation, but technologies like the Nobel Prize that was just awarded for CRISPR, we use that to make these mice that we study. And so these major fundamental scientific advances have helped us test exactly what Dr. Cook said. But what I would like to talk about is development. I think we know less about development than we need to know. A lot of our work is done in adults, and obviously autism, as you mentioned, is a lifelong disorder, but it has its basis in development, and that basis might even be prenatally as well. And that's something I think that is the real challenge, because you have a baby that is developing differently and gets diagnosed at 2.5, 3.5 years of age with autism, but the origins of that go back in development. And it's hard to use a retrospective scope to figure out what happened. And so that, I think, is the biggest challenge, to figure out how we can understand development better and then characterize it even when individuals are sort of later diagnosed. So that's the challenge. There's a quote from Wordsworth about the child is the father of the man, from one of Wordsworth's poems, and that is very true. I mean, the infant is the adult is the child of the infant in many ways, and we come from that, and so that's an important thing to think about.
So I think we're going to end on that. So we have some time for some Q&A. And the first question I have is for Suma. You mentioned, the prevalence is 1 to 54. I thought 5 or 10 years ago it was like closer to 1 to 100. I mean, do we have an epidemic on our hands? Why is it so, much more prevalent now than 5, 10 years ago? Are we diagnosing it better? Do we know more things? Suma, can you elaborate on that, please?
Yes, I think one thing is that we are better at diagnosis and also more consistent as we've developed better tools to diagnose. So more individuals are being picked up early and through school systems. So I do think that has been a dramatic change in some of the numbers that we're seeing. I can give you an example in Minnesota where we have the CDC data. When people are diagnosed more quickly in the Twin Cities area where they have more access to more resources than they are in the rural areas. So that would change. If you just looked at numbers, it would look different in rural versus the Twin Cities. But that's an example of where you have all that information and people are coming to get diagnoses sooner.
One positive point of this, I think, is that diagnoses can help, and they can help with access to therapies and treatments and education. That makes a difference in the lives of children and their development and the lives of their families. And so the one thing I see is that this increase in a diagnosis is partially because it actually is a value. There's been more acceptance of a diagnosis.
Well, we always talk about early intervention. Can you get in there early enough to cure autism?
I mean, one way I see it is the earlier you get in, the more chances you have to affect different brain pathways. So if you can do early intervention, some of the more rigid or restricted behaviors that develop when individuals have challenges communicating or functioning well can be worked on early. So I really see it as changing trajectories to improve functional outcome. So the earlier you are working with families on often critical period developmental stages. So Dr. Abel mentioned development is really important. There are times when learning to communicate is very important. And this early development allows you to get in there and give individuals different options to communicate. If it's not verbally, there are other ways with technology. And then that changes some of the other behaviors that evolve when an individual has challenges communicating.
So now let me switch gears a little bit. We're always talking about early intervention, which I just mentioned. As I said earlier, people are diagnosed, children are diagnosed, and then they grow up. And we're talking about finding new treatments through research. Will the treatments that you're trying to discover and identify help adults with autism?
So I'll tackle that one. We don't know. Professor Abel was very good at talking about the origins. The data point to the middle of pregnancy as to where many of the genes are expressed and are having an impact. And we don't know when we need to intervene. Many of the tests of some of the treatments coming out of mouse models, for example, and even fly models, one of the issues is we're not treating that early for concern about side effects in younger children. One of the genetic findings that's one of the more common ones is something called SYNGAP1. And in my clinic, I've had a patient I followed for 30 years, found out he had a SYNGAP1 mutation from some of the research that we've been doing. And working with another basic laboratory, the idea was to treat that with a drug. And in his 30s, there are benefits that we're seeing, such that his father said, well, wait a second, he's curious and learning to do new things, and we had kind of accommodated to not worrying about him learning how to use the key on the door as well, and almost wanted to put the genie back in the bottle. But it's a subtle effect. It's certainly the kind of effect I would also point out that would require much larger and careful trials, to be sure. But it's an example, and I want to talk about the biology a little bit more, that many of these genes, what we know is they're not involved in the overall development of the brain, but they're involved in very subtle timing in how nerve cells communicate with each other. And if we could alter that timing slightly in the adult brain, we may do a couple of things. And my first goal would be to help a lot of the patients who are very distressed by, let's say, their not being flexible with routines. And so I think the first thing we have to think about is making people's lives with autism more comfortable. I'm not sure that cure is really the goal here as much as helping people with their adaptation.
One of the things that we didn't cover are comorbidities or things like anxiety and depression, which can really affect individuals with autism. And especially if they're untreated in teenagers and adults, they can have a devastating effect. So that's just one of the pieces that could make a big change in outcomes for autism.
All right, I have one final question. Oh, and I think it's a yes or a no. It's that time of year for flu vaccines and hopefully a COVID-19 vaccine. Do vaccines cause autism? Ted?
No. No, they do not. There's no evidence to support that. In fact, the evidence that was published to support that was found to be incorrect and fraudulent from Dr. Wakefield in the United Kingdom. So the answer is a resounding no.
No, and I just want to say that most scientists and researchers are on the up and up every once in a while. There's one that, wants to get ahead, I guess, or publish things that aren't as accurate as they should be. So I just want to end on that and say, please go out and get your flu shot. I already got mine. And I want to say thank you so much for joining us. I think this was a really interesting conversation, and I hope everyone enjoyed it.
Thanks so much. It's great.
Thank you.
Bye-bye.
And thank you, Terry. See, I knew we would learn something important tonight. That was absolutely terrific. Thanks to all of our panelists. And thanks to all of you who joined us this evening. We appreciate it and appreciate anything you can do to help BRF on its critical journey. It's so important. Once again, you can contribute to this life-changing cause by going to thebrf.org/donate. Now, one good thing about this virtual world is that we can reach more people even after the event. So if you know of some friends or colleagues that you think would be interested in what you've just watched, let them know they can tune in anytime on BRF's website. Good night, take care, and hope to see you again next year, in person and virtually.